Va a esperar un poquito. Va a esperar un poquito.
Nada más va a presentar para
Ya.
que se vea bien.
Entonces ya lo estoy grabando.
Okay, yeah, so we can see your screen, so you can use your the full screen presentation.
No. No sé por qué hizo eso.
La última.
You're in the last slide, I think.
Yeah. I don't know why I did that. Vamos a probarlo otra vez. Ahora sí.
Yeah. Okay, so you just have, yeah, you just can close that one, yeah. Or yeah. So, all right. Yeah, because that's overlapping on your slide. So if you can.
We'll try.
There you go. So now we can see it. And it's running fine. So.
Yeah. Okay.
Okay.
Perfect.
So a couple of minutes just to introduce you. Okay. So.
Sounds good.
All right. So thank you very much, Henry, for giving us your time for this conference on migratory connectivity. So, guys, thank you so much for coming. And I wanted to introduce you to Henry Stevens. Henry is a PhD student at Georgetown University. He's about to finish his thesis. He's just in the last stage of writing his results and analyzing all the things that he got from the field. And today he's going to be talking to us about his work on this full-time job on his PhD that he has been doing over the last 4 or 5 years. So Henry, thanks so much, and we're ready for you.
Great, thanks for the invitation, Javier, and thanks for letting me talk to everyone. So yeah, Javier has invited me here today to talk about bird migration and migratory cognitivity in general, and then also talk about how I've integrated these concepts into my dissertation work. Y pues voy a dar la charla en inglés, pero si ustedes tienen alguna duda o no entienden algo o cualquier pregunta, avísame o avísale a Javier para preguntar a mí y podemos resolverlo, porque lo más importante es que todos entiendan lo que estoy diciendo. So, just a roadmap for the talk today. We're going to start with a background on bird migration and migratory connectivity. Then we're going to talk about how we typically estimate migratory connectivity. And then I'm going to walk you guys through a case study, which is just 2 of the chapters in my dissertation. The first focus on estimating range-wide migratory connectivity for a migratory bird species, the American redstar, that's Cephalophaga rickettsia. And then secondly, an example of how we can use range-wide migratory connectivity to understand historic patterns of breeding distributions for redstars. So without further ado, we'll start with a background in bird migration and migratory connectivity. So migration, which I define as a repeated annual movement in response to seasonal availability and resources or habitat, is a marvelous annual spectacle performed by hundreds of migratory bird species. And what better way to show migration than with my study species, the Cephalophila ricticea. And so here we're looking at the abundance of redstarts on the non-breeding grounds. In the spring, they shoot up to the breeding grounds where they have their young, and in the fall, they go back down to the non-breeding grounds. And these small songbirds repeat these journeys of thousands of kilometers year after year as long as they live. And it's important to note that migration occurs along a spectrum. And so different species may have like a preference toward one migratory behavior or another. But the more we study bird migration, the more we learn that really complicated even within the main categories of migratory behaviors we see. And so the first, and the one that we typically think about the most, is latitudinal migration. And this is when you have a bird's breeding distribution further north, and they move in mass to a more southern non-breeding population, basically along a latitudinal gradient. And so for most of the species breeding in the US and Canada, they're going to be performing some form of latitudinal migration. We also have different types like altitudinal migration. This is much more common in the tropics, where instead of moving along a latitudinal gradient, we have birds moving up and down in elevation. And so a good example of this is the three-wattled bellbird, which mainly occurs in Costa Rica and Panama. And you can see that during the non-breeding period, it's kind of at lower elevations closer to the coast, but then it has this really high elevation breeding period, which it moves to, typically following seasonal availability and fruiting resources, because three-wattled bellbirds are an example of a frugivore. We also have longitudinal migrants, which are a lot like latitudinal migrants. Instead of going kind of north-south, we see more of an east-west pattern. This form of migration is very common in particularly European species, but we also see it in the Western Hemisphere as well, with examples like the California gull that will breed in kind of these inland wet areas in the Rocky Mountain West, and during the non-breeding period will move westward to the coastline where it spends the whole winter. We also have eruptive migration, which is a particularly fascinating case of migration where, mainly because it's very difficult to predict. So in this case, these birds are not doing the same movements year after year. They're instead responding to seasonal availability in conifer crops, which are basically like little pine cones of different conifer species in the boreal forest. And so these conifer trees basically go on these boom-bust cycles in terms of how much cone crop they're producing. And in years when they're kind of in the bust stage of their boom-bust cycle, these birds have to migrate really far south in order to find enough food to survive the winter. And then there are birds that are becoming increasingly common as we study migration more and more that are actually performing multiple different migratory behaviors across their different populations. And so a great example of this is the American robin. And traditionally, we've thought about robins as being kind of a traditional latitudinal migrant like the redstart. And that's true for some populations of robins, like the ones breeding in Alaska. These guys are all going to migrate down to the southwest US during the non-breeding period. But as we track robins and study their migration more, we're finding that there are actually a bunch of resident populations scattered throughout this purple region of their range. So for example, where I currently live in Washington, DC, we know that the robins here are completely resident. And we also know that there are robins performing altitudinal migration as well, particularly along the Appalachian mountain range. And so you have one species that's performing latitudinal, altitudinal, and some aren't even migrating at all. But regardless of the migratory behaviors exhibited by a species, we're seeing that worldwide global change is threatening migratory birds much more than any other bird group. And so mounting evidence shows that migratory bird species are declining on average at steeper rates than other groups of birds worldwide. And on top of that, we know that more and more migratory bird species are becoming extinction prone through the years. And when we think about global change as a threat, we can think about threats at the individual level. And some examples of these are a migratory bird colliding with a window or getting eaten by a cat. And these individual level threats can scale up to impact populations, but we also have population level threats that affect migratory birds. And these are threats that act on very broad geographic scales, such as climate change. And so the combination of these individual level threats and population level threats scale up to impact migratory birds. And really the only way to understand the extent to which global change is impacting migratory birds is through the lens of migratory connectivity. And migratory connectivity is basically a fancy way of saying the degree in which individuals and populations remain together throughout the stages of their annual cycle. And to help illustrate this, I just have two basic schematics. So on one end of the migratory connectivity spectrum, we have an example of weak migratory connectivity. And so for this species, consider it has two breeding populations in Montana and DC, and two non-breeding populations in Mexico and the Bahamas. This is weak connectivity because the birds from these breeding populations redistribute equally among their two non-breeding sites. Conversely, at the other end of the spectrum of migratory connectivity, we have an example of strong migratory connectivity, where again, we have the same breeding populations and non-breeding populations, but because this species has strong connectivity, we see that all the birds breeding in Montana migrate down together to Mexico and back up to Montana, and the same is true for this DC Bahamas population. In other words, we see that these populations are linked. And this is overall an example of spatial migratory connectivity. And this is typically the way that we study and think about migratory connectivity. It's important to note that there's also temporal migratory connectivity, where instead of space, we're focusing on the timing of migratory events between stages of the annual cycles of these migratory birds. So the example of weak migratory connectivity now is for this Montana-Mexico population. We see some birds migrate on September 1st, some on September 15th, and some on September 30th. And so even though these birds are all in the same population, they're migrating at different times. Conversely, for strong connectivity, we have the two same populations, but we see that all the birds are migrating at the same time. And so regardless of spatial or temporal connectivity, you can imagine that the degree of migratory connectivity exhibited by a migratory bird is going to influence which threats it's exposed to, and in turn, how those threats scale up to impact breeding and non-breeding distributions and abundance of migratory birds. So let's say that we're thinking about redstarts again, and we see that they have weak connectivity, where birds from 3 different breeding regions are all migrating down to the Andes. In this example, any sort of large-scale threat like habitat loss or pollution or climate change is going to impact birds from all of these distinct breeding regions. But if we have an example of strong connectivity, in this case, strong spatial connectivity, we see that any sort of global change impacting the Andes is now only going to be an issue for these birds in one breeding population. In this case, Andes to Newfoundland. And then something happening in Mexico is going to only be an issue for these Northwest Canada breeding birds. And so the degree of migratory connectivity, in turn, influences the environmental conditions that a migratory bird experiences throughout its annual cycle. And to help illustrate this, how widespread this variability in environmental conditions is for many migratory bird species, these are pictures that I've taken throughout my dissertation studying American redstarts. And so they could be in the conifer forest of Montana, shade coffee plantations of Colombia, the deciduous forests of the Midwest US, the mangroves of Western Mexico. They really have to experience such a wide breadth of environmental conditions, and migratory connectivity determines the degree to which they are going to experience these conditions and the threats associated with the geographic regions they visit. And so when we think about migratory connectivity, I usually frame it in this kind of lens of understanding threats and population dynamics and seasonal interactions. But really understanding migratory connectivity is the baseline for answering any major ecological question about migratory birds. Because if you don't know where the bird goes throughout its annual cycle, there's no way to understand things like the level of disease transmission for different populations, or where to protect land to protect migratory birds, or even the amount of gene flow that dictates a population's ability to evolve in the face of global change. And so regardless of what your question is, understanding migratory connectivity is step one for any comprehensive study of migratory birds. And with that, I'll briefly pause for any questions that you guys might have.
Any questions right now? Right? No one so far, Henry.
Good. Great, so with that in the bag, now hopefully you guys understand a bit better about what migratory connectivity is and why it matters. And now I want to briefly go through how we typically estimate migratory connectivity. And so when we're trying to understand a bird's movements throughout the annual cycle, we can rely on intrinsic markers, which are any sort of biomarker that you can extract from the bird itself, or extrinsic markers, which are not related to the bird, but it's when we're putting something on the bird to learn about its movements. And so the first and really good example of an intrinsic marker to study migratory connectivity is stable isotope analysis. And particularly, we tend to focus on stable isotope analysis of hydrogen. And this allows us to estimate the mean breeding origin of any non-breeding population of migratory birds. And we can do this because of two main processes. Process number one is that we know that weather systems form in the tropics. And as they move north and all out of the storm clouds, the water molecules with the heavier hydrogen isotopes fall out first because they're heavier. And so as these storm systems move north, what ends up happening is that the ratio of stable hydrogen isotopes gets more and more skewed towards the lighter isotopes because they're falling out last. And so that's what these contour lines are showing. They're showing the ratio of stable hydrogen isotopes, and you can see that the values become more negative as you move north, because again, the heavier isotopes are falling out first, and the lighter isotopes fall out later. So that's process number one. Process number two is that we know that many migratory birds molt their tail feathers once a year, and this typically happens after they finish breeding. For redstarts, we know that this is typically around July, and that's pretty normal for most migratory bird species. And so when they're growing their new tail feathers, they're growing that tissue by using the commonly occurring elements in their breeding habitat. And so then if we go to the non-breeding grounds and we sample a tail feather from a migratory bird, we know it grew that tail feather in its breeding habitat. So if we analyze the ratio of stable hydrogen isotopes, we can therefore estimate the approximate origin of its breeding habitat. And so that's what so a lot of past authors have been doing, particularly over the last 15 years. It's become really common. This is an example for American redstarts, the study species of mine that I keep mentioning. And basically what the authors did here was go to all these different sites on the non-breeding grounds of redstarts and sample tail feathers. And then they were able to group the sample sites just based on geography and analyze those tail feathers using the analysis of stable hydrogen isotopes to estimate where the birds came from. And so, for example, their population A is this one sample site in Celestine. And using stable hydrogen isotope analysis, they were able to show that the breeding origin for these birds is likely in this northwest region of Canada. Likewise, if we say, look at population D, these two sites in the Western Antilles, we see that the analysis shows that these birds are likely originating somewhere on the east coast of North America. And so stable isotopes are a really powerful and relatively inexpensive way of estimating migratory conductivity. The other main intrinsic marker to estimate migratory conductivity is through genetics. And so what we're looking at here is a genoscape for American redstarts. And to build a genoscape, you first go throughout the breeding range of the migratory bird. So this is the breeding range of redstarts. And you sample tissue at as many sites as possible. So that's what all these black dots show. These are all sites where researchers sampled feather or blood. Basically, any tissue sample you can extract DNA from. And then they go through for each individual sample and they sequence its genome. And so from all your samples, you now have the genome of the individual. And you can cluster those genomes based on similarities. And so when we did that for redstarts, we see that we have these kind of 5 genetically distinct populations along the breeding range, where basically all the birds within one of these populations are very genetically similar to each other. And this is really valuable information because we can then go to the non-breeding grounds, and again, we can sample tissue and sequence the genome of the individual that we sample tissue from. And when we have that individual's genome, we can match it to the closest genetically distinct breeding population. And so with this genetic information, you can understand range-wide migratory conductivity. So for example, this red population, the maritime part of Canada, we see that if we sample tissue from redstarts on its non-breeding grounds, we see that these redstarts in the Andes are showing the same genetic signature as this population from Canada. And so that tells us that these populations are linked throughout the annual cycle. So to recap, isotopes and genetics are the 2 main intrinsic markers we can use to estimate migratory conductivity. But we're also relying more and more on extrinsic markers, which are effectively just tracking devices for migratory birds. And so as technology advances, there are just more and more different types of tracking technology being released to scientists. These include things like GPS trackers, which essentially give you real-time information on the exact coordinates of your bird. There's radio telemetry, where you put a radio tag on a bird and you can track it down with a radio antenna. Or you can use things like the MODIS system. And the MODIS system is basically this grid of radio towers that have been installed across North America. And if a bird wearing a radio tag passes by one of these MODIS towers, the MODIS tower will record that information, and the scientist will know, okay, my bird passed by this tower. But then particularly for small songbirds, because they're so lightweight, the only tracking technology we can use on them are these things called light level geolocators. And that's what I've circled here on the back of this redstart. And the geolocators are really simple, which allows them to be lightweight. They have the battery, which is this little bulb here. They have the light sensor, which is this little panel beneath the battery, and then a memory chip, which you can't see. And what geolocators do is they record light and time data. And from those light and time data, you can extrapolate estimates of latitude and longitude. Because at a given time, the amount of light everywhere in the world is slightly different. And so to show an example of geolocator data in action, this is a study from a postdoc in my lab who put geolocators on redstarts in a Jamaica non-breeding population. And he was able to get 12 back, 12 geolocators, and you can see you get the breeding estimates from those light data. So we see that the birds spending the non-breeding period in Jamaica are likely breeding in this Midwest region of the United States. And so combined, to estimate migratory conductivity, we have intrinsic markers like isotopes and genetics, and extrinsic markers like tracking technology such as these geolocators. And so I'll pause again there and, for any questions.
Anyone? Nope. Everything's okay.
Great. All right, so now that we've covered migratory connectivity and we know how to estimate it, I want to walk you through 2 chapters of my dissertation, where first I need to estimate range-wide migratory connectivity for my study species, the American redstart. And then I leverage that information on migratory connectivity to look into the past to understand historic patterns of distributional change for redstarts. And so to start with the first chapter, where we estimate migratory connectivity, this is essentially under the lens to understand how climate change has altered the breeding distribution of redstarts through time. And so in order to do this, we first need to understand what the current patterns of migratory connectivity look like for redstarts. And once we have that baseline, we can extend into the past, which is the second chapter I'll be talking about. And we can also look into the future, which is something I won't be covering in today's talk, but is another way to use this information on migratory connectivity. And so that's what I have here. This is the strategy I just talked about, where we basically need to understand the present so we can understand the past. And so to start off, I needed to understand range-wide patterns of migratory connectivity for redstarts. And luckily, going into this project, we already had the information on isotopes, and we also had information on genetics. So those are the 2 examples I showed for the intrinsic markers of understanding migratory connectivity. And really the last piece missing is range-wide information from tracking technology. And so again, since redstarts are such a small species, I was restricted to using these light-level geolocators. And so for my study design, I tried to cover as many distinct populations as I could across the breeding range and across the non-breeding range. So you see on the breeding range, I have a site in Newfoundland, New Hampshire, DC, Wisconsin, and Montana. And on the non-breeding grounds, I have Western Mexico, Jamaica, North Colombia, and South Colombia. And at each site, I tried to put out around 20 geolocators. And this is important because geolocators are archival. And archival means they store the data, they don't transmit it. And so in order for us to get those data, we actually have to deploy the geolocators, and then we have to return the following year. We have to look for the birds that survived and came back, and then we have to catch those birds, take the geolocators off, and extract the data. And I'd like to emphasize that this is a very time-consuming and difficult task. And so here's an example of us at our Western Mexico site. So this is in Cuyutlan, and so this is work that I did with Javier and his 2 students, David and Brian. And so this is us the year following the year where we deployed the geolocators. And so we had to canoe around the mangroves, trying to hunt down as many redstarts as we could to check and see if they were birds that had the geolocators on them. Because if they were, then we would try to catch them. And so doing that, all that field work took 4 years. And these are what the data look like combined. So what we're looking at here are the deployment sites. So again, these are all the places where I deployed geolocators throughout the breeding and non-breeding range of redstarts. And connected to the deployment sites, you'll see these dotted lines. So each one of these dotted lines represents 1 bird. The dotted lines do not show the migratory routes, they just show the linkages. So for example, take our Cuyutlan deployment site. We got 4 birds back, and we see the estimated breeding location from the 4 geolocators. So you see there's 2 in Colorado, and then there's 2 that bred more up in the Rockies of Idaho. And so you see these are all linked back to the Cuyutlan deployment site, just showing the linkages, not the migratory routes. And you'll see that each one of these points has this crosshair, and this shows the error of the latitudinal and longitudinal estimates. So you can see that some birds have a lot of error, and some have very little error. And this is simply due to the fact that the light data captured by the geolocator can vary between individuals. Sometimes a feather will cover the light sensor, or they'll be in like really dense vegetation, so they'll be kind of dark. So it's just variation in the light levels. And so you can see that we covered the range pretty well, with the notable exception being this kind of greater Yucatan area that includes the Yucatan Peninsula and then Central America, and then also this kind of more northern region of the breeding grounds. We weren't able to access this part of the breeding grounds just because there's not a lot of infrastructure there. And we did try to capture birds on the Yucatan Peninsula, but the density was not very high in the sites that we looked at, and so we were unfortunately unable to get data for those regions. But all in all, with the geolocator data, the genetic data, and the isotope data, if we combine these, we have a comprehensive understanding of where birds from every single population are moving during the full annual cycle. And this is really important because now we can use this information to understand how distributions have changed in the past and how they might change in the future. And I'll pause there for any questions.
Yeah, we have one question.
Perfect.
Hi, my name is Liliana, and I was wondering, like, what's the return date of the geolocators? How many of the birds that you mark come back with it, and do you mark them in any other way that you know maybe the bird is fine, but the geolocator failed at some point during the migration process?
Yeah, so great question. So the return rate depends on the exact population that we're studying. So for example, for the Cuyutlan population that we deployed geolocators on with Javier, we put out 18 geolocators and got 4 birds back. And so that's a return rate, I think, of around 20%. In other regions, we would get a return rate of like 30, 33%. And then also we've had populations like the Montana population where only 1 out of 20 came back. So it's highly dependent on the site. But the overall average is about what we saw at the Cuyutlan site, so like about 20%. So that's like 1 in 5 birds is coming back. And that's why we try to put out 20 geolocators at each site. That would ensure that we at least get 1 bird back, so we get some amount of information from the population. And in addition to putting the geolocators on the birds, we also give them a unique color band combination using plastic color bands. And so when we're back the following year trying to scout birds, all we have to do is look for color bands, because if we see a color banded bird, we know it should have a geolocator. And in my experience, all of the birds that we've recited with color bands have all kept their geolocator on, fortunately.
Thank you.
Yeah, thank you.
Anyone else? No? Okay, so that's it.
Perfect. All right, so that's the first chapter. We now understand range-wide migratory connectivity for redstarts. And now we can use this information to look into the past to understand how breeding distributions for redstarts have changed over the last century. And so for this chapter of my dissertation, I rely on this intrinsic marker of stable isotope analysis that I told you guys about in the earlier section of this talk, where we're relying on sampling feathers from the non-breeding grounds to estimate the approximate breeding origin where those birds came from. With that information, we can understand distributional change over time. And before I get into my own work, I want to give you some context for this project. And so my advisor, Pete Mera, has been operating this long-term non-breeding study of redstarts in Jamaica. And from these Jamaica studies, we've learned that redstarts are highly responsive to non-breeding rainfall, where in drought years, their body condition is much lower. And we also know from recent work that the mean breeding origin of these Jamaica redstarts has shifted southward about 300 kilometers in the last 30 years. And we learned this from a study recently done by a postdoc in our lab, Brian Dawson. And he, again, used stable isotope analysis to show this change in the breeding latitude. So remember, these birds are spending the non-breeding season in Jamaica. And if we look at stable isotope analysis of feathers taken from birds in the early 90s, we see this is their breeding origin. So this is kind of the Midwest area of Canada, kind of Ontario. And if we look at feathers more recently, from the last 5 years, we see the breeding origin has shifted south. And so that's that 300-kilometer southward shift I just talked to you about. And furthermore, Brian showed that the mechanism underlying this southward shift is drought conditions in Jamaica over the last 30 years. And remember, we know that drought conditions in Jamaica are bad for redstarts and reduce their body condition. And so after a year of really low rainfall in Jamaica, when it comes time to begin spring migration, survivorship for those redstarts migrating north is going to be lower. And it's going to be the lowest for birds trying to migrate farther distances, in other words, to more northerly breeding sites, because they don't have a good enough body condition to actually survive the journey. And so at a population level, this selects for redstarts migrating shorter distances, and this is why we see this southward shift in the mean breeding origin, because only those redstarts migrating shorter distances are actually surviving. And so if we zoom out and we think about this mechanism across the range of redstarts, and we assume that non-breeding rainfall is controlling the breeding origins for redstarts. And furthermore, if we think about the environmental variation in tropical rainfall throughout this whole non-breeding range of redstarts, we might expect that we see similar dynamics in the mean breeding origins for all of these other non-breeding populations. So again, for Jamaica, we know that the mean breeding origin has shifted southward over the last 30 years. But depending on the rainfall patterns in these other non-breeding areas, we could see similar or completely different trends. So for example, maybe it's gotten wetter in the Andes, and maybe we'd expect to see the mean breeding origin for these birds shift north instead of south because it's gotten wetter. And so we can actually test this across a long time period in the past by leveraging museum specimens of redstarts. And so remember, to do stable isotope analysis, all you need is a tail feather sampled from a bird on its non-breeding grounds. And so we have hundreds of museum specimens that were collected across the non-breeding grounds as far back as 1890. And so the tail feathers from those birds collected in 1890 is going to be representative of their breeding habitat during that time in 1890. And so luckily, since redstarts are widespread and relatively easy to find, they have good representation in museum collections across North America. And so what I did was I sampled tail feathers from a bunch of different museum specimens in these collections in Canada, the US, and Mexico. And I was able to perform stable isotope analysis on them to estimate their individual breeding origins throughout this last century of time. And so this is what it looks like in a lot of the museum collections. They just have a huge tray of museum specimens of redstarts collected from anywhere between 1890 to usually the 1960s. And all I did was I just would cut off the top 1 centimeter of the tail feather. And this is all we had to use to do the stable isotope analysis. And this is where the migratory connectivity information comes in. Luckily, from our first chapter, we know that the main non-breeding regions for redstarts are these regions that are circled here. So the Caribbean or the Greater Antilles, the greater Yucatan area, the Pacific slope of Mexico and Guatemala, the Isthmian region of Costa Rica and Panama, and then the Andean mountains of Venezuela, Colombia, Ecuador, and Peru. And so all I had to do was I had to group all of my feather samples into these geographic regions. And then furthermore, I grouped them by specific time bins to look at changes in the mean breeding origin through time. And so before we even look at the isotope data that I got from my analysis, we can actually make predictions about the directionality we expect to see for the mean breeding origins of these 5 non-breeding regions. And so, for example, we know in the Andean region over the last 100 years, it's undergone a drying period, followed by a wet period, followed by another dry period. And so since we're assuming that this mechanism of non-breeding rainfall is controlling the mean breeding origin, then we'd expect to see that mean breeding origin shift southward in response to the drought, shift northward in response to the wet conditions, and then shift south again in response to the dry conditions. And so I was able to go through and make predictions for all of the 5 non-breeding populations simply based on the non-breeding rainfall patterns. And so this is what we find. So I'm going to walk you through this figure because there's a lot going on. But essentially we have 5 panels. So each graph corresponds to one of those non-breeding regions I showed you. The Andes, Greater Antilles, the Isthmian, Pacific Slope, and the Yucatan. What we see on the y-axis is mean breeding origin as proxied by the ratio of stable hydrogen isotopes. So that's what this little symbol means. This just means ratio of stable hydrogen isotopes. And remember that the more negative the value, the more northern the breeding origin. And so that's what these arrows are here to show. They're just here to remind you that these values correspond with more southern breeding origins, and these values correspond with more northern breeding origins. In the orange, we see the mean breeding origin estimate for each one of our time periods, which are on the x-axis. So we have early 20th century, late 20th century, and modern. And you can see I put the years in brackets here beneath the time periods. And finally, the gray trend line shows the pattern of precipitation in that non-breeding region over this 100-year time period. So to walk you through the Andean population, we see that the mean breeding origin has shown a consistent northward shift through time. And we see that this is despite there being kind of this fluctuation in non-breeding rainfall. And so if we go back and we think about our predictions, how well did we do? We did pretty well. We were able to predict the trends for 4 of our 5 non-breeding populations, the Andes being the exception. So remember for the Andes, since it goes dry, then wet, then dry, we expected a southward shift, followed by a northward shift, followed by a southward shift. But we just saw a continued northward shift. So that did not meet our predictions. However, if we look at the Greater Antillian population, we see our prediction was correct. We see that the rainfall pattern here is primarily characterized by a period where it's got way wetter, and then a period of intense drought. And we see a corresponding northward shift in the breeding origin during the wet period, and then it shifts south again during this drought period. So this matches almost exactly what we predicted. And it shows that the non-breeding rainfall really does appear to be a mechanism controlling the entire breeding distribution of red stars. And so you can see that across populations, non-breeding rainfall was significant, but it appears to be probably stronger of an effect for these populations in the Greater Antilles and in like the Yucatan. And what's interesting is that all of these regions have significantly lower total rainfall during the non-breeding period. So just look at the y-axes of non-breeding rainfall. We see it, in the 2 regions in Mexico and the Greater Antilles, it never really goes above 50 meters of rainfall in a given year. Compare that to the Andes. we see that it's gone up to 150 meters of rainfall in a single year. And so what's likely happening is that these Andean redstarts are in super high quality habitat because of how wet it is all the time. And likely they're not as limited by non-breeding rainfall compared to some of these other populations. I just want to show you what the change in breeding origin actually looks like spatially. So here's a quick animation showing the estimated breeding origin, particularly specifically for the Andes population. So you see here, we start with early, late, and then modern time periods. And we see this breeding origin shifting northward through time over the last 100 years. And through our connectivity data, we know that these birds are likely moving their way up the East Coast. And I have geolocator data from the Newfoundland population in particular, and these guys spend their non-breeding period in this Andean population. So it's likely that these birds now breeding in Newfoundland, back in the 1890s, they were probably somewhere along the southern East Coast. And so the magnitude of these shifts in the breeding origin is incredible. And at scales that we didn't really believe were possible before. And so to summarize, in my first dissertation chapter, we used multiple data sources to estimate range-wide migratory connectivity for red stars. And that allowed us in the second chapter to understand how patterns of range-wide distributional change have developed over the last century. And in tandem, we can use our understanding of the present and the past to predict what future red star distributions are going to look like, say 50 or 100 years from now. This will be particularly important for many migratory bird species as we try to predict how they're going to respond to climate change in the future. With that, I'd like to thank everyone in my lab and involved in the field work, Javier for the invitation to speak to everyone, and then my funding sources, and I will take any lingering questions you guys have. Thank you all.
All right, thank you so much, Henry, for this very interesting topic. Anyone? Come on, Luis.
I have a couple questions if we have time. Yes.
Yeah, that's a very good question. So I did these calculations in terms of the total cost for everything earlier. I don't have the exact numbers, but one of those light level geolocators costs 120 US dollars. And so I deployed 20 of those at 9 sites. So that was 180 geolocators times 120, which I forget what that math comes out to, but it's in the order of tens of thousands of dollars. And of course, the travel to the US sites was not very expensive because I can just drive. But flying to Colombia and Mexico and Newfoundland and getting like housing costs, those add up quick. And so I think for all in all, my geolocator stuff probably costs 50 grand US overall. And if you compare that to isotopes, to analyze one tail feather, it costs 10 US dollars. And I analyzed 500 feathers. So that's 5 grand. But of course, you have to factor in the travel costs to go and get feather samples from all the non-breeding sites as well. So if you factor in the travel cost there, that probably brings that total up to about 15 grand. And then the genoscape analysis, I think, so I didn't do genoscape analysis. It was a collaborator, but I think he said if you incorporate all the travel to go to all those sample sites, because I think they had like 15 sample sites on the breeding grounds and like I think 10 on the non-breeding grounds, and then the actual genome sequencing, that comes out to like 100 grand total, 100 grand US. And so the isotopes are the most cost-effective strategy, but of course, what you save in money, you lose in resolution. And so the tracking data gives us the best resolution. We know like within more or less 100 kilometers, the exact location of the bird every single day for an entire year. And that includes during migration. Whereas the isotopes, you have the exact location of the non-breeding grounds because that's where you sample the tail feather, but the estimate for the breeding origin is regional at best, as you guys saw from the figures I showed. You can't get an exact like, you know, lat long for the breeding estimate. And then the genoscape is pretty good because you can relate the genomes of any sampled individual, and so that'll give you a fine scale estimate because it's just based on the sampling site. But of course, you lose the information linking the breeding and non-breeding grounds. So again, you don't have any information on actual migration. So yeah, so that's the breakdown for you.
Thank you very much. My other question is, from what I understand, the red stars are well represented within collections in museums.
Yeah.
But for species that are not well represented, usually how many years of data do we have?
Yeah, I mean, that's a good question. I haven't really looked closely at species that are data deficient. I will say that the kind of analysis I did of the museum specimens, I would typically not advise people to do that for rare species. And so since red stars are so well represented, me taking a little tissue feather sample from the specimen is generally considered to be okay because, you know, if they have tons of specimens and it's minimally destructive. But if you were doing this for like an endangered species like Kirtland's warbler, then yeah, I would not recommend this because they're so limited. The specimens are so limited in number that doing the destructive sampling probably wouldn't be advisable.
Okay, thank you.
Yeah, thanks for the questions.
Anyone else? Anybody else? Okay, Henry, it seems like that's it for questions.
Perfect.
Well, thank you so much, Henry. I think it's a very, very interesting results that you have. Very cool discoveries that actually I'm just learning too, especially about the predicting all these changes in the past, and I'm hoping to see what's going on with the future. And I know it's very interesting to see that shift in the breeding areas and how this rain conditions is affecting the distribution of the species in the breeding grounds. Yeah, so related to that, I just have one question. What is happening in relation to, we know that American wrestlers, they do show sexual segregation by habitat in the non-breeding grounds. And you mentioned that, at least for the Andes, it seems that they are wintering in good quality habitat so that they are able to go further north. And so what about considering this sexual segregation? What's going on in terms of like females that are segregated to lower habitat quality conditions?
Yeah. So yeah, great question. If you break down the shift in the mean breeding rate of those Jamaica red stars, the ones that are showing the lowest survival are going to be those females in immature birds that are getting relegated to the low quality habitat. And so particularly after a dry year, a lot of the adult males are not going to be as affected because they're able to stay in that high quality mangrove habitat. And so yeah, they're not gonna, their survivorship's not gonna be impacted as much. But we see the real drop in survivorship is for those females and immature birds that are getting relegated. And so yeah, so that's one of the underlying kind of breakdowns of those changes in the breeding origin we're seeing. And that would be true probably in other populations as well, although we haven't tested it. Like when we were in Mexico and then when I was in the Andes as well, we saw that segregation happening, whether it was mangrove habitat or in the Andes, the segregation usually looks like kind of the high quality riparian habitat near rivers and streams versus more like agricultural habitat. Yeah, regardless of what the breakdown is, it's the females and the immatures are always going to be the ones who have lower survivorship, or at least they should.
Okay. Yeah, no, that's interesting. So what about the situation? I actually was surprised to see that the the birds that are wintering in the Yucatan Peninsula are actually coming more from the western side of the North America. I didn't expect that really. I mean, I was thinking that they were coming more from the eastern or central areas of the breeding grounds, and so meaning that they are using like a different route of migration or going through along crossing the Gulf or going, you know, in the edge of the Gulf of Mexico. What's the situation?
Well, yeah, I would have really liked to have geolocators from that population to help answer that question, but we really have to just rely on isotopes and genescape for that. And so we don't know anything about their routes. But if I had to speculate, I would say they're probably using the same routes that we saw for the Western Mexico population, where it's going to be all overland migration. And they'll probably be migrating like through the Rockies because we know their breeding origin is, yeah, kind of northwest boreal Canada. And so they're likely just a little bit farther north than our western Mexico red starts. And they'll probably use that same western corridor and migrate down. And yeah, they probably won't do any crossings over the peninsula, if I had to guess.
Yeah, I would think so too. So, and my last question observation is that for the individuals that we uh, deploy the geolocators in Cuyatlan, and you show them that, you know, a couple of them are around Montana or Idaho in those particular areas, but within the map, it seems that they are south of the breeding, you know, area. So do you think that would be because the year that we deployed the geolocators, it was a really dry year, and they decided to go, you know, south or, you know, not too far in the north?
Could be, yeah. There's always the question of variation in the light data. I will say the breeding range polygon that I put on that map, that isn't necessarily the most accurate for the West. We do know there are little like kind of pockets in the Rockies,
Right.
where there are breeding populations. But yeah, it's also possible that there's probably this ongoing, there definitely is this ongoing trend in the Pacific slope population where we're just seeing this intense drought year after year. And so it's likely that the breeding origin for that population has been shifting southward, which is why we see our geolocator birds right at the fringe, kind of the southern border of that breeding range. And so yeah, it's hard to know exactly, but based on, yeah, my past isotope chapter, I would suspect that that's probably the underlying cause.
Yeah, for sure. All right, anyone else? Nobody? You sure? Last opportunity. Okay, Henry, I think everyone is now very
Everyone's
happy.
having a red start.
Yeah, no, but everybody's very happy to learn about your research. Thank you so much. I'm really looking forward to see the end of the story with your dissertation and the papers you published in the near
Yeah.
future. Thank you so much, Henry, and we'll keep in touch and say hi to people over there that we know each other.
Yeah.
All right.
Sounds good. Thanks,
Yeah,
Javier.
thank you so
Appreciate the
much.
invite.
Bye. Thank you.
See you, everyone.